Hello and welcome back, future scientists. My name is Mr. K. I'm here with Mississippi Public Broadcasting, and I've got a great lesson today talking about enzymes. Now, this is a little different than what we've been doing in the past because today I'm going to help y'all run a lab. Okay, this lab is on enzymes, and our goal here is to see how this enzyme performs under different environments. How does it perform in an acidic environment versus a basic environment? How does it perform in a cold environment versus a warm environment? Okay, that's what we're doing here today. So go ahead, start getting your safety equipment on, coats, gloves, goggles, whatever you got. I'm going to go over some of the directions and supplies, and then we'll be diving into this in just a little bit. So first of all, here are our supplies, okay? So one thing you're going to need are these 2-ounce cups. You can see them up here up front. I've got them labeled with some of the different environments that we're going to be testing. You're also going to need a potato. Teachers, you need 1 potato per class. What I did was I took a potato, I peeled it, and I put it in a blender with a little bit of water and made this potato puree. So that's what's in those 2-ounce cups up front, and that's what's in my little blender right there. Okay, you're also going to need hydrogen peroxide. That plays a big role in this lab. You're also going to need some kind of acid and base. Some really good examples of this is just vinegar for an acid and bleach for a base. Okay, use whatever you have, but those are some good options. You'll want safety goggles, safety gloves if you have them, okay? You're going to need eyedroppers, you're going to need tweezers, a stopwatch, and a beaker. And we'll figure out why we need all that stuff as we kind of get into the lab, okay? So here are kind of the directions for teachers and students, okay? So each student or group of students at your workstation, what you're going to need is that potato puree. You're going to need a pair of tweezers. You need a beaker full of hydrogen peroxide. That's what this is right here. That's full of hydrogen peroxide. You'll also need a stopwatch. And if you don't have a stopwatch, a phone will work just as fine. Okay? So what I did, like I said, I took that potato, I peeled it, I cut it into pieces. You just want to cut it up small enough where it'll actually fit in the blender. Put a little bit of water in there and then blend it up to make this puree. Okay, now the potato you will see will kind of start to turn this brown color. All that's going on is it's oxidizing. It's not going to affect these results, but also when you're cooking with potatoes, they'll turn brown like that. It does not affect the taste. People just find it unappealing to eat. So that's why you're usually dunking it in water to get the starch out and make sure that the potato doesn't oxidize and turn this brown color that people don't like. Okay, and then also here I have these little paper discs. Now I just got them out of a 3-hole punch, but you could also go to your copier and kind of empty out that paper disc tray and use those as well. Okay, so those are going to be some of the supplies that we need, and I have a little bit more directions before we hop into it. Okay, so what the teachers, make sure your students understand this. What we're going to do here is you'll take your tweezers, you're going to take one of these little paper discs, depending on what we're testing, you'll take that paper disc and then you'll dunk it into one of these different potato purees. We haven't set them up the right way yet, but then once you have them dunked in that potato, you'll take it and dunk it all the way into the very bottom of the beaker. What's going to happen is the enzyme that we're testing is called catalase. Catalase is in almost every living tissue. There's a lot of it in liver and potato, but from my experience, you definitely want to use potato instead of liver. That's going to help your room from smelling so badly. But what happens is as that catalase in the potato comes into contact with the hydrogen peroxide, the catalase is going to start to break it down. It's going to release some oxygen, and that's going to make the paper disc float as those bubbles are coming out of it. So what we're really testing is how long does it take for the little paper disc to float from the bottom all the way up to the top. We're going to time that. A quick time is a good reaction. That means the catalase was able to break that hydrogen peroxide down really quickly, and the longer it takes, that means the enzyme's not working the way that it's supposed to. So a short time is good, and a long time is not so good. Okay? So we'll start off with our controls, but then when we're testing the acids and the bases, we'll take a little eyedropper full of the bleach and put it into the base. I'll put it there so I don't forget. And then we'll take a eyedropper full of the vinegar and put it into the acidic to kind of get those pHs where we want to, and we'll do the same thing. Okay, so that's kind of the goal here. I'm going to go ahead and walk through it with y'all so you kind of know what's going on, but then I'd love to have y'all try to do this in your classroom as well, and make sure you have your data table here, because we definitely want to record our results when we're doing science, and I've got some review questions to go along with it. So if you're looking at your data table at the end, you'll kind of know what's going on a little bit better. Okay. Here's kind of a breakdown of what it could look like. This does not look exactly like what I have on the table, but just for teachers, this is kind of what you want. You can see there on the far left, that's a well plate. Those are the 6 different trials that we're going to run. We've got the different temperatures at the bottom and then the different pHs at the top, okay? And then there in the middle, that's kind of what the student's work table will look like. They take the tweezer, dunk the piece of paper into those different wells, let it soak up some of that catalase, and then again, you put the little piece of paper at the bottom of the hydrogen peroxide, and you're timing how long it takes to float up to the top. Okay, so we'll go ahead and get started with this. I wanted just to put my data table on the board back here so that I can record my results, and y'all can kind of see what I'm doing as we're going here, okay? So here's what it says. Using tweezers, you will dip a small paper disc into the potato puree. Okay, we're going to work with temperature first. So the one that I'm going to dunk it into to get started with is this one in the middle. It says room temperature, okay? So here's what I'm going to do. Again, I use my tweezers. I'm going to pinch off a little bit, just one single little disc. I'm going to dunk it into my potato puree, make sure I'm soaking up some of those juices. Again, inside of this potato is catalase, that's the enzyme that we're testing. Okay, I've got my timer here, so I'm just going to try to start it as soon as I dunk it all the way to the bottom. Okay, so now what I'm going to do, I take this disc, I'm going to put it into the bottom of this beaker, and then I'll start my timer and I'll stop it when it gets all the way up to the top. All right, are y'all ready? All right, let's see. All right, you can see it kind of floating up to the top. That one was pretty quick. I have it at just 4.86 seconds. Let's go ahead and round up with these. One of the things that I'm having us do is take the average. So let's just round up or round down to the closest number. So that one took about 5 seconds, okay? Make sure you're always including your units, 5 seconds, okay? All right, so that is the room temperature one. Now, I do want us to do 3 trials, though. When you're doing a lab like this, you want to do multiple trials. Data is weird, and so the more trials you do, the better overall average we're going to get, and that's going to be a little bit more true than if we just ran one. So I'm going to do the same thing 2 more times, okay? Let me take that one out. All right, so same thing, twice more. Okay, got a couple extra there. All right, so same thing, I got my little paper disc, I'm putting it into that room temp, let it soak up that catalase, reset my timer, and I'm going to do the same thing, put it all the way in the bottom, and then hit start when I release it, all right? All right. There you can see it floating up again. All right, that one was right on the money for 5 seconds. So trial 2 was 5 seconds again. Okay, here's one more, and then we'll move into some of those different trials, all right? All right. It's hard to grab these little things. All right, so room temperature again, soaking up the catalase, and same thing as before. All right, it's floating up to the top. All right, so that was 4.75, so 5 seconds again. I think I can do that average in my head. So 5 seconds, and the average there would be 5 seconds. So why is it floating up to the top again? Well, remember that This is potato puree. Inside of the potato puree is an enzyme called catalase. Catalase breaks down hydrogen peroxide. Hydrogen peroxide is H2O2. When the catalase breaks down the hydrogen peroxide, what you're left with is water and oxygen. And that oxygen is a gas, and it's wanting to float up to the top. So the faster it floats, the faster the catalase is breaking down that hydrogen peroxide. Okay, great. So that's my room temperature one. I'm going to put it out of the way just so it doesn't confuse me. So now what I'm dealing with is cold and hot. There's a couple different ways to do this, but in my experience, the results that work the best, instead of getting the puree hot and cold, it works a little bit better just to have hot and cold hydrogen peroxide. It's gonna do its job the right way either way. So what's going on is this is hydrogen peroxide that's been sitting in an ice bath, so it's nice and cold. And this is hydrogen peroxide that's been sitting in a warm bath. So it's a little bit warmer than room temperature. Okay, so what I'm going to do is pour this out, get it out of the way. And I think it wants us to do the cold one next, so let me just check. All right, so we'll do the cold and then the warm, okay? So I have my cold hydrogen peroxide here. It's been sitting in an ice bath. I'm pouring it in here. All right. I need just a little bit more to get it up to that 1 cup. Okay. So this will warm it up a little bit, but I think it's okay. All right. So, same thing. 3 trials, but this time we're dealing with the cold, okay? So let me take one of these little paper discs. Again, this was easy for me because I did just get them out of a 3-hole punch, but this is a really important part of the experiment. If you don't use pieces of paper that are the exact size, you would get different results here. So you either need to make sure you're cutting them out all the same size, or this is probably the easiest thing to do, is just to use a 3-hole punch and make a bunch of identical little discs of paper. Okay, so same thing. This puree really isn't different. It's the hydrogen peroxide that's cold. But same thing here, I'm taking that paper disc, getting some catalase in it, and this time I'm going to dunk it, same thing, and let's see what our results are with this. All right, so it's floating. It kind of took a little bit longer to get going there. And I know you can't see it from your perspective, but there are these little bubbles coming off of the disc. So it looks pretty neat as it does that. So this one was 8.85 seconds. So we're going to round up to 9. Okay. So this was 9 seconds. Okay. So definitely a little bit longer on that one. Let's see if this was a phenomenon or if it's a repeatable thing that we can see multiple times. So we'll run that back again. Same thing as just a second ago. Okay, little paper disc, little catalase, and then same thing as before. All right. All right, it's at the very bottom here, slowly floating up to the top. All right, that one was 8 seconds. So I think we are seeing here, compared to the room temperature one, this cold one on average is taking a little bit longer. Now we should know why that is. We've had a whole lesson on enzymes at this point, okay? Y'all might remember that the shape of enzymes are very important to their function, right? Well, what we're doing is we're changing the shape of the enzymes in the catalase. When we get it in a cold environment, that enzyme doesn't like the cold, and so its shape's going to change. When its shape changes, it's not able to break this down as fast. Y'all might remember that that's called denaturing the enzyme. So cold is definitely denaturing catalase. Let's do one more with the cold. Okay, a little bit of catalase. All right. All right, it's still at the bottom. It's floating now, though. It takes a little bit longer to get these going. All right, that one was 11 seconds. So that one was kind of longer than the other ones. 11 seconds. All right, give me just a second. This one's not quite as easy as the other one was, so let me get my calculator out. All right, so the average for this one was 9 seconds. So that's almost double what the other one was, okay? Okay, so I think we can see that the cold environment is not as good for the catalase as the room temperature was. Let's compare it to the warm hydrogen peroxide next. So I'm going to kind of dump some of that out. All right, put this out of the way so it doesn't confuse me. And then here's my warm hydrogen peroxide. It's been sitting in a hot water bath. So it's a little bit warmer than that room temperature was. Alrighty, so same thing here, y'all. We're taking paper, dunk it in the potato puree, timing to see how long it takes to float up to the top in that hydrogen peroxide. So, let's get started. All right, so here's my disc, catalase, sink it down to the bottom. We'll see how long it takes. All right, that one actually floated up really quickly. So that was only 2 seconds. Two seconds on that one, wow. All right, same thing, twice more. Goodness. All right, so little disc, catalase, let's get it in there. Also, if you are working with a partner, I'm finding it hard to start the timer and put this in at the same time, so that might be 2 jobs for the partner. Somebody's dealing with the stopwatch and somebody's dealing with putting this down towards the bottom, okay? All right, I'm gonna do this one over again. It didn't get all the way down to the bottom, so let me run that one back again just for posterity's sake. Okay, so let me give that another try, sorry. All right. All right, still very, very quick. This was 3.5, so I'm going to round it up to 4 seconds. So this one's kind of having the opposite effect as the cold water. The cold water was making it take longer. The warm hydrogen peroxide is actually making it go a little bit faster. So what that should tell us is that there's kind of an optimal range for this catalase. It likes to work at certain temperatures, more than others. Well, if you think about it, I told you all that catalase is inside of all living organisms, and think about our body temperature. Our body temperature is a little higher than room temperature. This one was a little bit warmer. This is closer to our body temperature. So that kind of makes sense to me why this one is going so much faster. It's kind of working in the range that it likes to. All right, last one for the temperature. I got the catalase on my paper disc. It's going so fast I barely even have time. This was 2.4, so 2 seconds on that. And the average for this is 2.6. So let's round up to 3 seconds. Okay, so 3 seconds. So what we're seeing here, like I said, it's the same enzyme, it's the same hydrogen peroxide. The only difference in between these is the temperature of the hydrogen peroxide. and we're getting different numbers. We're getting different times and averages on how long it takes. The cold one took a little bit longer, the room temperature was kind of in the middle, and then this hot one, the warm hydrogen peroxide, we're getting the best results here. So the catalase inside of this potato is working best when it's in this warm hydrogen peroxide, okay? Great, so that's one part of the lab. The other part I wanted to do has to deal with pH. So let's give that one a look. All right. So, kind of the same thing here. The majority of the lab is the exact same, okay? We are taking these paper discs, putting them in hydrogen peroxide, seeing how long it takes to float up to the top. This time, though, I'm going to change the pH of our potato purees a little bit. So what I want to do here first is, this is vinegar. Vinegar is an acid, okay? So I'm going to take that vinegar, and I'm going to take a couple of eye drops full and put it into this acidic puree. And that's going to lower the pH a little bit, okay? Okay, I'm going to mix it up so I'm not just getting all vinegar. Okay, it's very important when you're doing this. Let's use different eyedroppers for the vinegar. So that was one for the vinegar, and now I'm going to do one for the bleach, okay? We don't want to use the same one. That's called cross-contamination. It's going to mess up your results a little bit. So I have a different eyedropper. Same thing, I'm going to take a couple of eyedroppers full of bleach and put it in to this puree. All right? And same thing, let's kind of mix it up so I'm not getting all bleach here, okay? Okay. Y'all know probably that bleach can mess up your clothes, so it's very important. Try not to spill it or get it on anybody. It could take your nice pair of pants and ruin them, so be careful around that. Okay? And then I'll fill up my beaker with some hydrogen peroxide. Great. And I'll do the same thing. So, alrighty. So, little paper disc. I'm going to dunk it. Let's start. This is telling us to start with the acid. Okay, so I'm going to take the acid, going to mix it around, and same thing here. Let's put it into this hydrogen peroxide and see how long it takes to float up to the top. All right, it's sitting at the bottom here. Okay, so this, it's not moving. Now this is not bad, okay? What we're gonna do is let it go for 1 minute. If nothing happens in a minute, that means that this catalase and this is not working, and that's okay. That's part of what this lab is supposed to show. What's happening here is that this vinegar, this acid was so strong that it's denatured or broken all of the enzymes in this catalase, and now it's not working anymore. It's not floating up to the top. I don't know if you can see it, but it's right there. It's really not doing anything, okay? So we're gonna let this run for a minute. If nothing happens, we're just gonna record it as a minute. All right, y'all, that's 1 full minute, so to me, it doesn't look like we're going to get any reaction out of that. So to me, it looks like that this enzyme, the catalase in this potato puree, might be denatured. But let's do a couple more trials just to confirm that. So here's another one. We'll do the same thing, see if we can get any reaction out of this, okay? Okay. So let's run that back again. I can barely hold on to it. All right. They're right next to each other. You can see there, not really any reaction going on. So where this catalase didn't really mind the temperature too much, it could work at a cold or a room temperature, and it really liked the hot, it doesn't seem like it can really handle much of a pH change. Okay, again, that's called denaturing an enzyme. We're breaking it where it doesn't work anymore. All right, well, that first one was 1 minute, and the second one also looks like it's not doing anything, and so we're going to write 1 minute on that one as well. Okay, let's do one more and just see if we can get any type of reaction out of this catalase. Okay. What do y'all think? I don't think it's going to do anything. We'll let it run just in case. But this catalase does not like acidic environments very much. The other ones were really just shooting up there towards the top, because as that catalase breaks down the hydrogen peroxide, it's releasing oxygen, and these little bubbles were kind of forcing it up. But here, the catalase isn't working, so there's no bubbles, there's no oxygen, and it just sits there at the bottom. All right, y'all, I let it sit there for a minute, but it didn't do anything, so I'm going to go ahead and write 1 minute here, okay? I think I can do that average in my head. That one's going to be 1 minute. Okay, great. So we've already done the room temperature one. We'll test this neutral out too. This is basically another control group, but just to show that this is supposed to be doing something. Let's run the neutral a couple of times, and then we'll end with the basic one here in just a little bit, okay? So, taking a little paper disc. Let's dunk it in that neutral. So this is just the potato puree. There's no acid, there's no base in here, and this is just regular hydrogen peroxide. You see how fast this one's floating up to the top? That one was 3 seconds. So that's a lot faster than the acidic. All right, let's give it another couple of tries. In science, you want to do as many trials as you can. That's going to give you more data, and more data is always good. It's going to give you a more accurate picture of what's really going on. Okay? All right, that one was 4 seconds. So this should be working. There's no reason why the acidic shouldn't have worked other than that vinegar was too much for the catalase to handle. So here's our last neutral trial. So paper disc, right in there, dunk it in the bottom, and let's see if it does anything. It's floating up a little slower than the other ones, but that was still 4 seconds. Great. Okay, so I don't have anything written down on this one because we kind of already did a control, but I just wanted to show you that there's no reason why it shouldn't be working. Great. All right, so really great. Let's go ahead and try this basic one, and then we'll see what our results are. And I've got some questions to go along with it. So same thing, little paper disc. If the vinegar didn't work, I'm really interested to see what this basic one does. So swirling it around, and let's do the same thing here. It's just sitting at the bottom. It's not moving. All right, I'll let it sit there just in case. But I'm really feeling like it's only going to be 1 minute. All right, y'all, it's not making any moves here. To me, it looks like that catalase has been denatured because of the bleach. It's been a minute, so I'll go ahead and record that. it's not really able to do anything with that. Okay, so let's run this back twice more and see if we can get any type of reaction to happen. Okay. Also, I probably should have mentioned this earlier on. This one's not really doing anything. This is just hydrogen peroxide, so it's okay if you get some on your hands or fingers or whatever. A lot of people use hydrogen peroxide to like heal and clean out a wound, so it's totally okay if you touch it a little bit. It's really the bleach that you want to avoid. Okay, it's just, oh, actually, this one is floating a little bit. I don't know if you can see it. It's bobbing up and down. It's about right here. It was rising for just a second. There it goes again. So we might actually get a reaction out of this one. Look at that. That is all the way up at the top. That's great. So that one was 40 seconds. Okay. Again, so this is not really working anywhere near as well as the neutral pH or that warm water, the warm hydrogen peroxide that we did earlier, but this one was working. So that's telling us that the hydrogen peroxide, or sorry, the catalase inside of this basic puree is not totally denatured. There are some enzymes there that are still working. Okay, so same thing, last trial, little paper disc inside of that puree. We'll dunk it all the way in the bottom here. Let's see if we can get a result out of this one. I don't know. Maybe we just had a lucky odd phenomenon there. This one doesn't look like it's doing anything either, but we'll just watch it to make sure. Okay, it's 30 seconds, still no reaction, but let's just stay with it just in case. All right, we're going to give it a little bit longer. It rose, it stood up, but it didn't really, it's not really rising. All right. All right, that's 1 minute, so I'm going to go ahead and stop it and put 1 minute here, okay? Okay, let me find my average for this one. So the average here was 53 seconds, okay? So yeah, what this lab is supposed to kind of show is how enzymes react to different environments. And we got good results here today, because what we were able to tell is that this catalase, again, this is potato, inside of the potato there's catalase. The catalase really liked warm hydrogen peroxide. That probably gets the hydrogen peroxide a little bit closer to our own body temperature, right? And so it works really well under those environments. It did okay at room temperature, and then it didn't do great in cold hydrogen peroxide. Okay, so it does have an optimal temperature. It likes it more warm, doesn't really like it cold. And then we moved here to the pH, and we kind of did something similar, but instead of working with hot or cold, we're working with acidic and basic. It really hated this acidic environment, okay? It almost didn't have any reaction. That means that all of the catalase inside of this little container is denatured. It's not working anymore, okay? The neutral one did okay, just like we thought it would with the room temperature. It was very similar to that, maybe just 4 or 5 seconds each. The basic, we kind of got some interesting results there. It did not like the basic environments, but it liked it more than the acidic environments, because we did get one that was able to float up all the way to the top. All right, everybody, thank you so much for hanging in there with me. I hope y'all learned something out of that. I had a lot of fun doing that lab with y'all. Your teacher has some other assignments for you to kind of work on, so make sure to give that your best effort, and I hope everybody has a great day.